For the organizers to allow me to present this time. Also, I'd like to really quickly acknowledge my team or my advisors, Dr. Maribel and Dr. Pita, as well as my close collaborator, Ana Machos, and my fundings, which are there. And well, I promise this will be a short talk. Well, my project is about the inference of horizontal gene transfer from an evolutionary perspective and using graph theory. So in particular, you know, graphs consist of dots and edges connecting those. Those edges are connections. And in particular, we use relative divergence time connections. So I will explain what is that. I don't know how to move. It's not working. Okay. Okay, so this is a classical example provided by you and collaborators on how important is horizontal gene transfer. In this example, plants evolve from marine medium to terrestrial medium thanks to a lot of gene transfers coming from bacteria, archaea, and other kingdoms of life. Well, you know, here everybody loves bacteria. In this slide, I'm presenting you a lot of mechanisms that bacteria uses to transfer genetic material. You can use extracellular vesicles, you can construct like bridges between bacteria, or even virus can help to these transfers. This is the motivation for us because inferring the evolutionary history of horizontal gene transfer in bacteria might be very complicated because there is a lot of transferences. And this is related to the problem of antibiotic resistant genes, which is very dangerous for us. Okay, so very quickly, I will bring some definitions. I will recall some definitions. We have a species tree. We use trees to model evolution. So basically, evolution is a branching process, and well, in the leaves of the tree, we have a species for a species tree. Also, we have gene tree, in which the leaves are genes, and well, genes evolve inside the species, so we embed gene trees inside the species trees. And we also assign evolutionary events. We have speciation, duplication, gene loss, and well, don't forget about horizontal gene transfer, in which genes are jumping between branches of the species tree. And well, the classical way is that genes are inherited from ancestor to descendants, but in horizontal gene transfer, genes jump. Okay, so this is the bioinformatics challenge. We can observe species, we can get genes from those species, and well, we represent the genes as sequences. So the question is how to go from sequences to evolutionary scenarios and horizontal gene transfer. Okay, so I promise you to some gene-to-gene relationships, and particularly relative divergence time. So this is like the key concept behind our approach. So basically, we can estimate the divergence time between species, which is like represented by the height of this bifurcation. And also, we can estimate the divergence time between genes. And well, we can now have these questions. Does the genes diverge at the same time as the species? So this is like a null hypothesis. Genes diverge at the same time as the species, but also genes can diverge prior the species or later than the species. Okay, so we are going to estimate divergence times. But maybe Sue can complain a little bit here, or the lion, because this is really difficult to infer times of divergence. But the one robustness of our method is that we don't need precise estimations. Sometimes precise estimations are impossible, but we only need to know somehow statistically if the genes diverged before, after, or at the same time as a species. Okay, so now I will present our approach, the graph tripartition. So let's assume that Shala and collaborators provide a theoretical characterization. So we assume that we know the evolutionary history. In other words, we know a gene tree embedded inside a species tree. And well, based on this evolutionary scenario, we can construct a graph like this one, which is called graph tripartition. This is a graph where everything is connected with everything, but we have three different kinds of connections. That's why it's called graph tripartition. So I will disentangle this a little bit. I will start with the EDT, equal divergence time, part of the graph. And basically, we will connect two, so in this graph, the nodes are the genes, and we connect two genes if they diverge at the same time as the species. As a quick example, we can look at A1 and A2. They diverge at this point of the species tree, which is the speciation. So then we connect A1 and A2. Another example, maybe A5 and A1. They also diverged at the same time as the speciation. So they are here, A5 and A1 are connected. So this is one part of the graph tripartition, which is called equal divergence time. Also, we have the prior divergence time for genes that diverge before the species, and we have the later divergence time for genes that diverge after the species. Okay, so as you see, we reconstruct this graph from the evolutionary scenario, but we don't know the evolutionary scenario. We want to reconstruct it. But we can estimate these relationships also from data. So as this is a quick example, we have, let's assume that we have a graph, an LDT graph with the genes, this species, and only one edge. And we can theorize what are the possible evolutionary scenarios that explains this graph. Since we have three genes and three species, we have three possible gene trees, three possible species trees, and 9 possible combinations. We do all the combinations and check when we obtain this graph. And we discovered that there is only one gene tree topology in which C is the outgroup that explains this graph. So if you don't believe me, here I have the 9 examples. We can review them later. Well, the message from this slide is that you, if you look like locally to parts of your graph, you can obtain pieces of the gene tree and the species tree, and then you can merge all that together to reconstruct the whole evolutionary scenario. This is called informative triples. You listened this term yesterday in Maribel's talk, but in this case, they come from data for horizontal gene transfer instead of best match graphs. Okay, so we implemented already this methodology. So we have genomes, real and simulated. We estimated distances using our tool, Revolution tool. Then we have some Python code to approximate the later divergence time graph using a statistical approach. And well, this is statistics and we are prone to errors. So we have to ensure that these, the graphs that we are inferring make sense mathematically and evolutionarily speaking. So we use an integer linear program to correct the graph and ensure these mathematical properties of that were shown in previous characterizations. Okay, so this is the statistical approach. Basically, we have this line regression line. We have in one axis gene distance, in the other axis species distance, and everything that lies in the regression line is considered equal divergence time. The outliers are or LDT graph or PDT graph, PDT relationships. Well, this is just a linear regression. It's nothing new for anyone, but actually it's very difficult to deal with linear regressions when you have a lot of outliers. So we apply 3 different methodologies for this, and also we apply the Cook distance for the outlier detection. Also, I told you that we correct the graphs using an integer linear program approach. Basically, we define a system of equations and then we have to minimize some numbers in order to have like an optimal solution. So here are some numbers. Remember, we want to infer gene-to-gene relationships that are supposed to come from an evolutionary scenario. We simulated data, so we know what are the true relationships. And well, let me show you some precision results. Like in general, our precision is very high, close to 1. This means that if we predict an LDT relationship or a PDT relationship, it is very likely that our prediction is true. And well, we tried several methods, and some of them perform very well. An example, the IQR performed very well for PDT, but very bad for LDT. So we will have to mix the methods somehow. Well, as a summary, well, everybody loves bacteria. Horizontal gene transfer is a key process in species evolution, and our method ensures the existence of an underlying evolutionary scenario explaining our predictions. And this is like a very, very important thing, because tons of methods do similar things, but they don't ensure that the predictions actually fits in an evolutionary scenario or in evolutionary theory. Okay. Future directions, we will explicitly reconstruct the evolutionary scenarios, not only the graphs. We will apply our methodology for metagenomes from this beautiful land. And well, we have to improve runtimes, basically getting rid of the ILP approach because it takes too much time. It might take forever. Thank you.